So you're now familiar with what an amino acid is, and you know that in order to build a protein, you've got to figure out how to make covalent bonds between one amino acid and another, and another, and another, and so on. You want to make a big chain of amino acids. That's going to be how you chemically build a protein. So let's take a look at how you might do that. I'm going to use a generic amino acid picture like that, and I'll redraw it. You can redraw it in the box down on the bottom of page 17. I'm going to use my marker board for this so that I can draw and erase and move things around. So on the left, I'm going to draw an amino acid. For this illustration, I don't care which one of the 20 it is. I'm just going to draw any old amino acid. Whoops, I'm going to draw it correctly. Sorry. I'm going to draw any old amino acid, and because I don't care which one it is, I'm just going to put an R down there instead of putting real atoms. So in blue, there's an amino acid. I'm going to draw a second one next to it. It's going to look just like it. It's going to be a separate standalone amino acid. Here's my amino group. Here's my central carbon. Here's my carboxylic acid group, my side chain, and that hydrogen on the central carbon. So here's two amino acids side by side. Right now, they are two completely separate molecules, independent. Every atom in each one of those molecules is perfectly happy the way it is. You can look at any one of these blue atoms and it's forming exactly the right number of bonds. Again, think back to a couple videos ago, we talked about how to draw these structures, and we talked about bonding rules. We said that every carbon, when it becomes part of a larger molecule, needs to be forming exactly 4 bonds. Well, 1, 2, 3, 4, 1, 2, 3, 4, etc. We said that every oxygen has to be forming 2. Here's 2, here's 1 and 2, etc. We said that every nitrogen has to be forming 3. 1, 2, 3, 1, 2, 3. And that every hydrogen has to be forming 1. There's 1 and 1 and 1 and 1. So these are all okay. In other words, there's no atom on either of these pictures that thinks it has an incomplete valence shell. They're all perfectly happy the way they are because they're bonding the way they're supposed to bond. If I want to get somebody over here in blue to covalently bond or share an electron with somebody over here in red, I'm going to have to take bonds that already exist, disrupt them, and then rearrange electrons and put things back together again. Let me show you what that looks like. Let's say I were going to, let's say I could reach in and pick up one of these molecules. Of course, they're too small to literally do that. But let's say I could sort of grab onto that oxygen right there, and I could grab onto that carbon right there. Right now, they are sharing a pair of electrons. That's a covalent bond. That's represented by that bar right there. But if I separate them enough, if I get them far enough away from each other, they can't share anymore. Think back to a video that we talked about a couple weeks ago with covalent bonding. Remember the snowblower and the lawnmower. People get too far apart, they can't share anymore. Same thing here. If that oxygen and that carbon get too far apart, they can't share anymore. So let's go ahead and do that. Let's drag that oxygen, the hydrogen's still attached to it, it's going to go along for the ride. Drag that far enough away from that carbon where they're not sharing their electrons. The oxygen keeps its electron, the carbon keeps its electron. No longer do you have a sharing of these two electrons, which was the bond. You just broke a bond. So you've got a mostly intact amino acid with an OH missing, and separate from that, you've got an OH. More importantly, you've got a carbon with an electron that it would like to share, but isn't. You've got an oxygen with an electron that it would like to share, but isn't. Keep that in mind. I'm going to move this down a little bit here. Keep that in mind, and we'll go ahead and look at the other amino acid. Let's say, again, that I could reach in here and grab onto that nitrogen and grab onto one of those two hydrogens. Let's pick this one. And I separate them. I get them far enough apart so that they're not able to continue to share their valence electrons like they were doing here. They were making a bond. They're not anymore. If that's the case, that hydrogen's going to fall off, so to speak, leave. It's going to take its electron with it. The nitrogen is left with its electron. This used to be the shared pair, used to be the bond, but no bond anymore. They're too far apart. So now, instead of having two completely intact, perfectly happy amino acids like I had in the beginning, I've got four separate things. I've got this almost complete amino acid with a nitrogen looking for something to share with. I've got this almost complete amino acid with a carbon looking for somebody to share with. I've got a hydrogen atom, and I've got a hydroxyl group. That's a functional group we talked about before called a hydroxyl group. So let's say I shuffle these things all around here, and I let them rearrange themselves so that this nitrogen gets really close to that carbon. Now, I'm not going to erase it and redraw it, but imagine that these two, the red and the blue, get very close to each other. Close enough so that that nitrogen's electron sort of has some interest in that carbon nucleus, and this carbon's electron sort of has some interest in that nitrogen's nucleus. In other words, these two atoms, that carbon and that nitrogen, could get close enough so that they can actually share this pair of electrons. If that's the case, well, that's a covalent bond. What I just drew here in black ink is a new covalent bond. Now, again, if you want to make this a little bit neater, erase that and redraw it really close, but you get the idea. There's a covalent bond right there. Before we go any further with that, let's look at what happens to these other parts. You've got a hydroxyl group, an OH. That oxygen's got a spare valence electron. You got the hydrogen atom looking for something for its electron to do. Put those close to each other. Let them be shared. What did you wind up with? It's a molecule of water. So what did I just do? I took two separate, perfectly happy, perfectly content, standalone amino acid molecules. I broke a bond off of here, I broke a bond off of here, and I rearranged the parts that I got when I made the breaks. And I put them back together again with new covalent bonds. That resulted in a molecule of water hanging around. Fine. This happens in cells all the time, and cells have lots of water in them, so one more water molecule on the pile doesn't matter to anybody. But more importantly for this discussion, this results in the attachment with a covalent bond between one amino acid and another. And that was our goal, if you recall. When we started this, I said, how are you going to attach an amino acid to another? How are you going to get this to covalently bond to that? Well, that's how. You're going to break stuff off and then rearrange, kick some parts out. I kicked out a hydrogen, I kicked out a hydroxyl group, kick them out of the way so that you got a free electron here and a free electron here that can then be shared. And oh yeah, as a side effect, water formed. Who cares? Nobody cares about that. That's fine. This is what I care about. I now have two amino acids covalently bound to each other. Now, that's not big enough to be called a protein yet, but what if I took a third amino acid and I put it over here somewhere, and I did the same kind of reaction? Now I'd have three. And if I did that again and again and again, maybe I'll have dozens or even hundreds of amino acids strung together, one to the next, to the next, to the next, by doing the exact same kind of reaction over and over and over again. Each time I do that reaction, a molecule of water gets kicked out of the way, and I get this to happen. That's how cells build proteins. They grab an amino acid, they chop a piece off, they grab another, they chop a piece off, they make a bond. Then they grab a third amino acid and chop pieces off and make another bond, and then a fourth, and a fifth, and a sixth, etc. Each of these reactions, ones that we just summarized here, can be called an example of a dehydration synthesis reaction. You see that on page 17. And this type of reaction is super important in chemistry in general, but certainly in biochemistry. We'll see other examples of it going forward. But for right now, this is a specific phrase to refer to the type of reaction that occurs between amino acids when you join them together this way, when you're building a protein. Why dehydration? Because what does dehydration mean? It means to remove water. Well, I removed parts from here that happen to equal a water molecule. Why synthesis? Synthesis means I'm building something. Well, I'm building one big molecule out of what used to be two separate molecules. This is now one big, I'll call it a double amino acid if you want. That's one molecule now. It used to be two separate molecules. So that's a dehydration synthesis reaction. One last thing on here. The type of bond that formed from this carbon to this nitrogen, I called it a covalent bond, and in fact it is. You can see it's the sharing of a pair of electrons. But you can give it a more specific term or name if you'd like to. Sometimes it's helpful to do this. That specific covalent bond that you see right there is often called a peptide bond. And that's in the key terms box at the beginning of this section of the lecture outline book. A peptide bond is simply the covalent bond that exists between one amino acid and the next amino acid in a chain. Well, when you chain a bunch of amino acids together, you've got a protein. That's what proteins are, a big chain of amino acids. Just a little bit of root word here, PEP or PEPT. If you see that in a word, you know that word has something to do with proteins. So to call this a peptide bond, it's kind of a giveaway. It tells you, oh yeah, it's got something to do with proteins. It must exist between amino acids, because bonding amino acids together is how you get proteins. So it's correct to call it a covalent bond, but it's more specific to call it a peptide bond. And that's the basic chemical explanation of how proteins are formed in cells. Again, we've just done one of these, but do this a bunch more times, dozens of times more, hundreds of times more, and make a big long chain of these amino acids, and you've got a protein.